I listed some of the mechanisms of peripheral tolerance that we discussed last time. So if you recall, I told you that there were 6 mechanisms of peripheral tolerance. For each, we've been talking about both how they can get made and how they can get broken. And we talked about clonal deletion, helplessness, suppression, and deviation last time. I left off talking about some details relating to deviation. And so I want to finish what I have to say about deviation, and then I'll go into the final 2. That's why there's 2 little blank spots up there. So if you recall with deviation, clonal deviation really has to do with the balance between different helper T cell subsets. And so the idea is that what will happen is that typically when we are making some kind of T cell response, there is a particular type of CD4 T cell helper subset that will be better, be sort of protective, and one that might be worse. And we usually will generate the cytokines to make to polarize those T cells into the good kind of helper T cells. That's clonal deviation. We're basically pushing the cells away from being the bad kind. And if you remember, clonal suppression was making regulatory T cells. And in some ways, you can imagine deviation and suppression being similar, given that you could deviate towards or away from the regulatory T cell response. I told you one part of the application of this last time, which was the hygiene hypothesis. But I want to show you a couple of other sort of examples to think about this info. So this paper actually shows kind of an idea of how people think about this actually working in real life. So what you can see here is you can see 2 different mice. One mouse is a wild type mouse, and one mouse is a mutant mouse. So it's missing a particular gene, ATG16L1. The thing that you should see from this is that there is a genetic component in this disease situation. However, just because the mouse has this particular mutation doesn't mean that it ends up with Crohn's disease. In this experiment, we also see mice that have either wild type phenotype or this mutation that are getting a particular infection with a particular virus. And this does not show you a sort of additional example of what happens to the mouse if the mouse doesn't get this virus ever. And the answer is it lives happily ever after and has the normal phenotype as well. But here we can see the mouse getting different versions of a virus. This is a murine norovirus. And so you can see that wild type mice, it doesn't really matter which version of the norovirus they end up with. They are sort of have long-term the normal phenotype. If these mutant mice get one type, they will also have the normal phenotype. But if these mice get a different version of the virus, that may push them to a Crohn's-like disease. And so what you should notice here is that there is a role for genetics and a role for environmental exposure. So it's sort of like this mouse had to be doubly unlucky. Actually, I guess triply unlucky. It had to have this gene, it had to get infected with a norovirus, and it had to get infected with this kind of norovirus instead of this other kind of norovirus. All of which were leading this mouse to Crohn's disease. And this sort of relates to this idea of deviation. You will see more about this on the next couple slides, where there was a similar model that has been described that we understand in even more detail. One other thing that is important about this particular piece of information, so this shows you the citation for the paper that this information came from. you guys have a literature assignment. And so for the lit assignment, there is a paper that you're going to read. And there are also 2 kind of news articles talking about the same phenomenon. I love that paper. And actually, one of the news articles is better than the other, but I couldn't not include that other one because A, it's pretty short, and B, it's funny. So it made me sad to think about not including it, because then you wouldn't get the funny part. But in sort of the longer news article, it talks about labs, other labs that have been thinking about similar kinds of projects. One of those labs is the lab that did this paper. And in fact, this paper is sort of part of that literature as well. So if you're reading that and sort of reading about some of those other labs, this is actually a piece of that. But the model that we understand a little bit better about deviation is the model that is shown in this study, and there have been a number of follow-ups, largely coming from the lab of Terence Dermody. And so this shows a situation that potentially can lead to celiac disease. And so first, we can look at what's going on the left. Okay? And you can see that we start with a dietary antigen. We can talk about the details of some of the specifics of celiac later. But now, really, all you need to know is there's an antigen that you ate. As we discussed last time, typically the dendritic cells and macrophages in the GI tract tend to be really good at inducing regulatory T cells. So we have this phenomenon of oral tolerance, where it tends to be that when you encounter an antigen first in the GI tract, you end up having tolerance to that. And so normally, in a person without celiac disease, they ingest the antigen. A dendritic cell picks up that antigen, presents that antigen, and makes cytokines such that the T cells that are responding become Tregs. And then we are tolerant to this food antigen. So that's what this normally should look like. And in fact, this was sort of an example I told you about for clonal suppression last time. However, now this is on the right-hand side. If a person is really, again, there's a fair amount of unluckiness in this. This person is infected with a particular strain of a virus. In this case, it's a virus called reovirus. And it's not any reovirus. It's just the red one, T1L. It's not the greenish one. So it shows the greenish one, and this doesn't happen with the greenish one. But it does happen with the red one. So you have to have sort of be very unlucky enough to the first time, so that the first time you ever have this antigen, you also happen to have reovirus at the same time. So if you happen to be really unlucky, now some dendritic cells are going to be getting the food antigen, and some dendritic cells are going to be getting the reovirus antigen. Reovirus is a microbe that infects the GI tract. And those dendritic cells are going to generally send out cytokines in the area. This is going to be a different sort of mix of cytokines going on in this area than it would have been otherwise. And as a result, this T cell is not going to learn how to be a Treg. It's not going to get the Treg cytokines. It's instead going to get a different set of cytokines. And now it's going to become a Th1 cell when it should have become a Treg. And now that it's a Th1 cell, it can actually cause some damage and can lead to celiac disease. And so this is all coming from this sort of very unlucky event where our person just so happened the first time they ever encountered this antigen, they also had reovirus the same day. And they also happen to have this strain of reovirus. And you can see this here as well, that we can have our antigen, reovirus, both coming into the dendritic cells. Now the dendritic cells are going to make cytokines that push the T cells to Th1 cells. They should have pushed them to Tregs in the case of the dietary antigen, but they're not, because they're actually getting the PRR stim from the virus. We make a different set of cytokines, our Th1 cells, and now we actually make these Th1 cells that can go back and start to damage the intestine and lead to celiac disease. And so what you can see here is that by sort of switching the helper T cell subset that is generated in response to an antigen, we can switch the disease state. And if we could figure out, for example, a way to make sure that these T cells didn't become Th1 cells, if we could block this part of it, we could deviate them away from making disease. And so this whole model for celiac disease has been worked out pretty nicely over the past 5 years or so. Yep?
Does it ever happen in the other way, that there are too many Tregs and not enough?
Yes. So does it happen the other? So Sebastian wants to know if it could happen the other way, that you could have a situation where you bias a whole bunch of cells to Tregs, where it shouldn't be the other things. You could. If you remember when we talked about the cytokines that bias to different subsets, some of the inflammatory subsets like Th17 are the Treg stuff plus. So it's easier to go the other way because you get like added things onto the Treg stuff. But yes, there are examples where you end up biasing to Tregs that you shouldn't have. And then you usually end up kind of not controlling the microbe and having a lot of damage from the microbe. All right, so that was clonal deviation. So now we need to go into our fifth mechanism of peripheral tolerance. And just like the 4 we've seen thus far, number 5 is really super related to other things we've talked about this semester, and in some ways will look really obvious. Because the fifth mechanism of peripheral tolerance is called clonal anergy. And you guys are well aware of anergy. In particular, when we think of clonal anergy as a peripheral tolerance mechanism, we often think of it with T cells, because B cells often go to anergy as a central tolerance mechanism coming out of the bone marrow. And what you can remember is that a T cell needs to see signal 1 and signal 2 in order to be activated and proliferate. And if that T cell only sees signal 1 without signal 2, that T cell will become anergic, and that T cell will not be able to be stimulated later. So you can see all of that happening on this slide. And so how we make anergy is pretty straightforward. So this, and the reason why it's sort of interesting is that there are a few ways that we are aware of that anergy can go wrong. And many of them we understand kind of, and we can imagine them leading to autoimmune diseases pretty easily. And so a lot of the things I'm going to tell you about, at least in terms of stuff we understand, are kind of ways that autoimmunity can happen. And so there are 2 different ways that we can see anergy happen. One of them is shown here. This one sometimes people get a little confused about, and people want a fancy name for it, and there isn't always a super fancy name for it. So I apologize if you want like the term that means this. There isn't always a term. One thing that does make me super excited is that earlier in the semester, someone asked me a question, and that question was basically this. So you guys have already figured it out. So hooray, nice job, team. And so at the top, you can see a situation where we could get anergy happening. And so what you can see in this situation is we can see a time where our dendritic cells are presenting self-antigen. And what I want you to remember is that we are always presenting self-antigen. 24/7 we are presenting self-antigen. So here you can see a cell presenting self-antigen. So it is going to show that T cell signal 1, but there are no PRRs around, and so there's nothing to provide signal 2. And so that T cell is going to be energized. But now we can imagine a situation where we have infection. And in a situation where we have infection, we now have PRR ligands that will activate our dendritic cell so that our dendritic cell will start to make B7. And so you can see the B7 in purple here being made by our activated dendritic cell. But when you think about that dendritic cell, you shouldn't think of it as having one copy of MHC class 1 on its surface showing off a peptide. You should think of it as having lots, like hundreds, thousands, lots, okay? Even more than to the 2 that this figure shows you. And all of those MHC molecules that are presenting on the surface of that cell are not presenting the same peptide. They're all presenting some peptide that they picked up. It's a peptide that binds to that MHC, but they're not all going to be the same. Usually, the frequency with which they are presented is related to how frequently that peptide is made. So like if there's one peptide that there's a million copies of in the cell, it's probably going to be presented pretty often. If there's one peptide that's kind of rare in the cell, it'll be presented less often. It's like going to be sort of an idea of that. The ones from the microbe are probably going to be pretty frequent. You're probably going to have a lot of copies of them. And so the majority of the MHCs on the surface are going to be presenting stuff from the microbe, but not all the same ones. They're going to be all different peptides from throughout that microbe. And I'm making this number up, but imagine that like 90% of the MHCs on the surface of that dendritic cell are presenting something from that microbe. It used to be 0%. Now it's 90. There still are some MHC molecules that are going to be presenting self-antigen. Because it's not like we have no self-proteins in this cell. We still have self-proteins. They're still getting degraded at some rate. Some of them are still going to end up on MHC. And in the event that this sort of situation happens, the B7 protein and the MHC protein are not actually like bound to each other or linked. They're diffusing randomly in all different directions around the surface of the cell. And if there is a T cell with T cell receptor and CD28, that will engage both of them. And so this dendritic cell is going to present some microbe peptides to a T cell that responds to microbe peptides. And it's going to also have B7 on its surface to interact with CD28. And that's going to induce this T cell response to the microbe. And that's what we want. That's kind of like how it all's supposed to work. But it's also possible that we could have a situation where a self-peptide is getting presented and pairing with that same B7. because this cell is now making a bunch of B7. It's just throwing it on the surface. And this could stimulate a self-reactive T cell. If you happen to be unlucky enough to have a situation where a self-peptide gets presented and just happens upon the T cell that's self-reactive, that could activate that self-reactive T cell and lead to autoimmunity. And so now we would have turned on this autoimmune T cell. Yep, Jamie.
So if you're always presenting some degree of self-antigen, how does this not cause like vast amounts of autoimmunity?
Yes, and so we can look at this and we can say, oh my gosh, this should be happening all the time. We should be breaking tolerance all the time. And this goes back to a piece of information we talked about regarding memory cells. Both a pro and a con, okay, on the sort of memory cell info. So I'm going to tell you something that's going to seem unrelated, but then we're going to come back to, I'm going to turn it back around, okay? So I want you to think about what's going on in this autoimmunity situation first. In this autoimmunity situation, there's no microbe, right? There's no infection. These, whatever tissue they are, I don't know what they're trying to make that tissue be, whatever tissue that is. It has no microbe. It's not making any B7. So how come this T cell can work and destroy this self-tissue? There's no B7 in this case. So why do these T cells work now? Yes, Sebastian.
Because after they get the co-stimulatory signal from the injury cell, they can see the
Yeah, so T cells only need to see B7 the first time they are activated. Only naive T cells need signal 2. Once a T cell is activated, it doesn't need signal 2 anymore. Its threshold for activation is lower. And so basically the idea is these T cells happen to get B7 the first time they ever see antigen. And then as a result, now they're active, they're on. They are going to sort of kill here because they don't need this anymore. So now let's think about flipping this to the question that Jamie asked. We've got a situation where we have a T cell that is self-reactive. When do you think that T cell first encounters its self-antigen? So you got a T cell, self-reactive, comes out of the thymus self-reactive, going to respond to a self-antigen. When do you think that happens? Yeah?
I mean, it depends on what the self-antigen is.
Depends a little bit on the self-antigen, but let's imagine the host, the person in whom this is happening. When in the person's life is this most likely happening? Relatively young. Or it's probably happening relatively soon after the T cell gets out of the thymus, right? Because you're making new T cells every day. So you made a T cell right now. It might be self-reactive. It's probably going to find its self-antigen pretty soon. What's the most likely scenario for that T cell to find its self-antigen? What's the most likely situation where that T cell finds its self-antigen? Probably on some healthy self-cell, right? If that happens, you get this. So most of the time, the first time that T cell sees antigen, it's going to be seeing antigen without co-stim. And it's going to be energized. And then the situation that we just saw doesn't matter because that cell is energized. And now, even if it does see antigen along with signal 2, it stays energized and stays off. And so the problem in this situation is this T cell is seeing antigen for the first time. And it's seeing antigen for the first time with co-stim. And so again, if you think about probabilities, this is a relatively low probability event. And if that happens, then that T cell can go into an autoimmune situation. The alternative is that you have to figure out a way to break anergy, which really usually happens with just boatloads of cytokines. Does that make sense? Okay. So that's one kind of big piece of how we can break anergy. And you can imagine the ways that this can be kind of similar to what we might see in a autoimmune situation. The other mechanism is something known as molecular mimicry. And this kind of ties back into how a T cell is seeing peptide on MHC. So remember, the T cell is not seeing the whole protein. It's seeing a peptide of, we're talking about a CD8, 8 to 10 amino acids. And it's not only is it seeing only 8 to 10 amino acids, it's not really even seeing all 8 to 10. Some of them have R groups that are now buried into the MHC pocket. So really, the T cell is only seeing kind of like the top of that peptide along with the MHC. And so there are some situations where there will be pathogen peptides and self peptides that have enough similarity that they can actually kind of look similar as far as the T cell is concerned. And so here you can see the peptides aren't actually identical. There are even some examples where they are pretty much identical. But they're similar enough that if you were a T cell, you might get confused. If now this T cell gets activated by the pathogen that it responds to, and so you can see a situation where we are making a response to the pathogen. This is not showing signal 2, but there would be signal 2 in this situation. That T cell is now going to become an active T cell. It no longer is going to need signal 2. It's going to stop being dependent upon signal 2. And so now, later on, if that T cell goes out into the body and finds that self-peptide that is a mimic of the pathogen peptide, suddenly that T cell can cause damage. A lot of this tends to be related to specific MHC types because only a particular MHC type will be presenting a self-mimic that looks like a pathogen peptide. And so only certain MHCs will be presenting peptides that have the unluckiness of looking this similar and allowing this whole business to start. And in that case, you would have to have that MHC type and you would have to get infected with that pathogen in order for this whole business to happen. And so you can see there's a luck part of this. This is showing this process as a sort of schematic. Here you can see the same kind of general idea. Perhaps there is some respiratory infection, but it just so happens that one of the antigens in the respiratory infection is also an antigen that's found on the beta cells in the pancreas. If that happens, then T cells are going to go in and they are going to kill those beta cells in the pancreas, thus leading our person to have type 1 diabetes. One other piece of this that we understand is that sometimes we can get some mimicry to happen if the T cell receptor binds to the MHC plus peptide at the wrong angle. So typically, what we see is shown on the far left in A. So the blue is the MHC, the green is the peptide, and the yellow and red are the TCR alpha and beta. And so you can see the TCR kind of is going right across the middle of the MHC plus peptide at this characteristic angle. These are two examples of autoreactive T cells and how they are binding to MHC plus peptide from multiple sclerosis patients. And so here you can see the T cell kind of is binding weird off to the side. Here you can see the T cells binding at totally the wrong angle, totally off to the side. And so it does seem like perhaps some piece of this may have to do with sort of weird binding reactions. In addition, all of these examples of molecular mimicry that I've shown you are T cell examples, but this is not limited to just T cells. This phenomenon actually talks about a disease, an autoimmune disease that happens that's antibody molecular mimicry. Particularly in this case, there is a bacterial infection with a particular type of streptococcus. And individuals make antibodies against antigens on the cell wall of that type of streptococcus. One of those types of antibodies actually cross-reacts with an antigen that's in the heart, which you can see here, leading to damage to heart muscle. And this actually causes a disease known as rheumatic fever. And so the moral of this story is it's not just an antibody cross-reactivity thing. It also can be a T cell. It's a T cell cross-reactivity thing. It can also be an antibody cross-reactivity thing. And what I hope that you sort of are realizing from all of this is that there does sort of seem to be this role for infection in autoimmunity. You could imagine in the molecular mimicry case, one would have to be infected with just the wrong pathogen that might be cross-reactive and lead to their autoimmune disease. You can think about in that breaking anergy situation that there needed to be infection with a pathogen. The other thing I've mentioned is that you could imagine that a cell might stop being anergic, and this is actually sort of experimentally hard to deal with, so I don't have a great figure for you. I've told you before that anergic cells can become activated if they get like a sledgehammer of a signal, right? Really, the idea is just so many cytokines, so many. That situation can happen when somebody is dealing with an infection, particularly a really bad infection. They can have so many cytokines that some of their cells that were previously anergic could get turned back on. And in all of those cases, we can see situations where infection might predispose someone to autoimmunity. There's usually a situation where you have to have the particular type of microbe and a particular HLA type. These associations are really hard to make because usually what happens is you might have the infection. I'm making this up. Imagine you have that infection when you're 5, right? You have Coxsackie A when you're 5. That presents as cold number 73 in your kindergartner. You're probably never going to get it diagnosed as Coxsackie. You're probably going to go home and drink orange juice, right? And then what happens to you as a 5-year-old is you turn on one bad T cell. And that one bad T cell is going to take some time to expand. Might take a few years because it's not seeing antigen very often. Might take a little while. It's going to take some time to expand. It's going to take some time to actually start killing beta cells. And it's going to be a while before it kills enough beta cells that you actually get sick. And so you might not start to have symptoms until you're 15. It might have taken 10 years between when you turned that T cell on and when you actually show up at the doctor's office and are like, hey, I have diabetes, And when you're 15 and you show up to the doctor's office, you're not going to say, you know what I think? I think this is related to runny nose number 76 I had as a 5-year-old. Like, you're not. In fact, you're most likely not going to ever be able to make that tie. Your doctor is not going to be able to sort of tie your current autoimmune disease to a previously undiagnosed infection. And so there are some ways that people have done this and have been able to make some of those things. And they're also probably not going to go as a first round of testing you for HLA as well. And so generally, we can sort of imagine how this can work. And for most of our understanding of autoimmune diseases, this is sort of what is happening. But being able to say, in this person, it was this infection, in this situation, we pretty much can't do. And so we usually think that there is a big role for some kind of infection predisposing or sort of breaking tolerance. But that's pretty much as specific as we can usually get. That reovirus example I showed you before is one of the few examples where we know a little bit more about it. All right, so what questions do you have for me about this and breaking anergy? Because breaking anergy is a big one. Okay. Now we get to do number 6. And number 6 is a fun one. Number 6 always causes much drama. So the final mechanism of peripheral tolerance is also related to some things we've learned about before, but it is not as kind of obvious from stuff we've learned earlier in the semester as the others. So like anergy, you're like, yeah, we learned anergy. This one is sort of a little bit more out there. If we try to use the same kind of naming conventions we've used for the other types, then we would refer to this one as clonal ignorance. It is also often referred to as immune privilege. Immune privilege is often discussed in relation to the organs that are listed here. You can see they are listed as the immune privileged sites. And so we can kind of think through immune privilege and what might be going on with immune privilege. So if you look at those 4 organs that are listed here, what do those 4 organs have in common, particularly evolutionarily? Like particularly think about, if you were going to think about sort of what might be important with these organs, or what do they all have that's important about them evolution-wise? Don't overthink it. Yeah, Rishi. So what do you mean by what do you mean by survival? Yeah, all of those things are things you really don't want to mess up. And all of those things, if they got messed up, that would be a serious disadvantage evolutionarily. You want to evolve to protect those things. And if you sort of rank things you need to protect if you want to survive and have offspring, those are probably the things you need to protect the most, right? You start destroying your brain, bad things are going to happen. Start destroying your eyes, bad things are going to happen. Obviously, you start destroying testes or the uterus, you're not going to survive and reproduce because, well, duh. Right? So these are organs that really cannot take damage. The classic understanding of immune privilege. I'm going to tell you the classic understanding, and then I'm going to show you some experiments that have made us realize that classic understanding, it's maybe a little messier than that classic understanding. The classic understanding of immune privilege is that in the immune privileged sites, we exclude adaptive immune cells. So adaptive immune cells are unable to attack in those areas. So you cannot have damage. So they cannot mistakenly damage things. The idea is it's just too important to sort of make sure that these body locations are okay. So we actually exclude the adaptive immune response. Another way of saying it is that we actually hide these things from the adaptive immune system. So there's no way that they can get damaged by some immune response going wrong. One of the places, or one of the things that we talk about with the classic understanding of immune privilege is a disease called sympathetic ophthalmia. And so this is sympathetic ophthalmia. One fun fact about sympathetic ophthalmia, it's not a very fun sounding disease as I tell you about it, but one fun, in quotes, fact is that this is actually how Louis Braille went blind. So what can happen? So the idea here is that there may be a situation where someone has trauma to one of their eyes. And this is like you get stabbed in the eye kind of trauma. To the point where you actually are breaking membranes behind the eye. And suddenly eye antigens are released from the eye into the vessels behind the eye. And so the idea is that the eye used to be hidden from the immune system. And now it is no longer hidden. You've taken these antigens and you've broken them out so they can suddenly be seen by the immune response. And those antigens will end up going into the nearest lymph node. And when they go to the nearest lymph node, they turn on T cells, who are like, whoa, what is this new antigen I never saw before? This must be something real bad. I must be infected with some terrible thing. Oh no, got to get rid of this terrible thing I'm infected with. And those T cells head back to the eye and completely kill it and destroy it. But not only do they destroy the damaged eye, they also destroy the non-damaged eye. Even though you got stabbed in one eye, you go blind in both eyes. Because T cells are now like, oh my gosh, look at this whole area with this antigen. And in fact, you end up losing both eyes. And so this is sort of the classic example for immune privilege, and thinking about the eye as the immune privilege site. I'm going to tell you in a second about a bunch of ways that immune privilege gets a little wonky, or a bunch of ways that maybe not as clear, as clean cut as I'm describing it here. But one thing that we do know quite well is that in many autoimmune diseases, there is a phenomenon known as epitope spreading. And epitope spreading, conceptually, is similar to immune privilege. So it's not always, it doesn't have to happen in an immune privileged site, but it is a concept that is similar to that classic understanding of immune privilege. And so the idea with epitope spreading is that you might have a self tissue, and you might have a situation where you might, say, have a self cell die. So here, our self cell is going to have this virus, this infectious agent, and we might kill some of those self cells. When we do that, we might release some other antigens that were previously not accessible to immune cells. You can imagine them as being hidden. Now it's not the same kind of anatomical barrier of like the back of the eye. Now it's just the anatomical barrier of, say, being in a cell. but you can release more antigens, which can now prime a new set of T cells. And now we're going to see, instead of responding to one antigen, we're going to see responses to many. And so we've spread out the number of epitopes we've responded to, epitope spreading. And so now we've kind of amplified autoimmune diseases. This can happen in a case like this, where the microbe leads to suddenly these proteins being no longer sequestered, being seen. There can also be situations where if you start making a response to one self-protein, as you destroy cells, suddenly you uncover new self-proteins and make new responses, and so your autoimmune disease amplifies. And so you get autoimmune amplification. And so conceptually, this is similar to immune privilege, though it is, again, on a slightly different level. But there are some problems with immune privilege. So if you look at what I'm telling you here about immune privilege, what's the obvious problem with the classic model of immune privilege, as I told it to you? Usually I tell people about immune privilege, and there's like, Dr. Barker, there's an issue here, yeah.
So you were saying for immune privilege, basically that those areas don't have an adaptive response, which should protect them against any sort of autoimmune disease, but it's not going to protect them against like pathogens.
Yeah, these are the super important tissues, and we leave them unprotected from pathogens?
Yeah.
What?
Sure.
Seems a bit of a problem, right? And so that's one. So there has been kind of evolving understanding of immune privilege for a long time. But one of the places where this has become kind of interesting has actually been related to Ebola. And this is a slide that shows you a bunch of past Ebola epidemics. And the thing I want you to see on this is kind of, so these are actually different Ebola epidemics over time. And the numbers are the number of people who were involved in each epidemic. And you're seeing both the fatalities in the darker color and the survivors in the lighter color. And so you can see that in this situation, or in this figure, the biggest ever Ebola outbreak involved 425 people. And we'll say about 200 of them survived. You can see that there aren't a whole heck of a lot of survivors in most cases. And you can see that, we're talking the big outbreaks are like 400, 300. That was true until 2014. The 2014 Ebola epidemic in West Africa, this is actually went from 2014 to 2016. These are the totals, which is that there were 28,646 people who were infected and 11,323 who died. So you can see the numbers are way bigger. Way, way, way bigger, right? In addition, you will see how many Ebola survivors there were, right? There are, in fact, more Ebola survivors currently alive than there ever have been before. Because usually, we had relatively small outbreaks. There were relatively few survivors. Now we have this huge outbreak, and we had some treatments in some cases, so now we have this huge group of people who are all Ebola survivors. And there had been weird anecdotal stories about if you survive from Ebola, weird things happen to you for years. But there just weren't very many cases of this to actually see if it was real or not. Now suddenly we have a whole lot of people who've survived Ebola. And let me tell you about some things that have happened. So this is one doctor who was treating patients with Ebola. This is what his eye used to look like. And this is what his eye then changed to look like. And so you can see that he basically had major changes to his eye. And that's because it turned out that the virus stuck around in his eye. and caused damage in his eye, even though he thought he was completely cured and the virus was no longer in his blood. And if we look at some of the other stories about some of these Ebola survivors, some of these Ebola survivors end up with eye problems, loss of hearing, sleep difficulty. All of those are either eye or brain things. Joint pain, abdominal pain, back pain, fatigue, impotence, severe headaches, memory problem, confusion. Basically all brain and eye things. And this has been, data on this have been published. And we've also seen there are some cases of Ebola being sexually transmitted. In some cases, up to a year. Well, the understood cases are up to a year post-infection. You think you're cured, you have no virus in your blood, and then you transmit to someone else. There are some suspected cases that are 5+ years that are being heavily debated right now. And so, and in general, we see that these were all cases of, they were all cases actually of male Ebola survivors transmitting to their partners, or pregnant women transmitting to their partners. I was going to say all male, and then I was like, no, there was that one case, but it was a pregnant woman. And in fact, so there have been all these situations where the Ebola outbreak was thought to be over, and then suddenly it was like back. And that was because of transmission from, sexual transmission from some of the survivors. This woman, Pauline Cafferkey, I feel so bad for Pauline Cafferkey. She was a nurse who went to West Africa to help Ebola patients. She got Ebola 3 times while she was there. She basically, and the reason why was because it seemed as though the virus, she got infected, she cleared the virus, and she had the virus still in her body in immune-privileged sites, and it kept coming back out. And her story is very sad. Just not a good time. I would not want to be Pauline Cafferkey. I would not wish any of her story on anyone. And so what we realize here, if you look at all of these situations, is that these Ebola survivors are having problems with eyes, brain, reproductive organs, and you were seeing the virus persist in some of these immune-privileged sites and come back. And so first of all, the question of, is this potentially problematic in immune-privileged sites is yeah. You know, is there a downside to this? Yeah, there is a downside to this. But the other place where we really have started to understand this differently is in this case. So we started to sort of question some aspects of immune privilege here as well. And the other case that I'm going to tell you about is a case that is related to a particular disease, multiple sclerosis. And so you can see some general details of multiple sclerosis here. The idea with multiple sclerosis is related to this at the bottom about nerves. So what you can see is that usually nerves have this myelin sheath surrounding parts of the axon. You can kind of think of that myelin sheath as being like plastic on a wire. That helps make sure the electrical impulse goes in one direction instead of shorting and going off in other directions. And so it's basically keeping the conduction going from one end to another. In multiple sclerosis, you lose some of that myelin sheath. And so you can see you have demyelination. The myelin sheath is attacked, so you have holes in it. And then nerve conduction does not go linearly in the way that it should. You get little shorts. That's really, Dr. Knowles could explain it much better. However, that is kind of what we need to know right now, is it's about loss of myelin. In the case of MS, what we know is that there are T cells that make their way into the brain. those T cells actually are actively going to lead to the destruction of the myelin. And so the reason why the myelin is destroyed is largely T cell related, though there is also some antibody pieces to this, but we'll say T cells for now. And so something happens. We don't really know what the something is. As a result, T cells go into the brain and start destroying the myelin sheath. And some immunologists came up with a potential treatment, an idea of how to deal with this. And they said, well, so the problem here is that we have T cells going into the immune privileged site. Shouldn't normally have T cells going to this immune privileged site. So what we're going to do is we are going to treat people with an antibody. And that antibody is going to bind to some of the integrins on T cells so that those T cells can't go to the brain anymore. So we got this problem. T cells were going to the brain and destroying things. The idea is, well, T cells aren't supposed to go to the brain. If we just block the integrins, then we fix immune privilege. We reprivilege the site. I just made up the idea of reprivilege. We reprivilege up the site. T cells are no longer allowed to go into the brain. They're no longer allowed to leave the vessels. So we all live happily ever after. And you can see this is actually the number of new lesions in a person with MS. You can see that they generally, it really was revolutionary for these MS patients in terms of stopping lesion formation. It was great, wonderful for these patients. And then it wasn't. Because all of these patients, so this got approved in 2004, and about a year later, these patients started having these pretty severe brain issues in a disease called PML, or progressive multifocal leukoencephalopathy. And they tried to determine what was going on in these patients' brains. Why were these and PML actually has some weird cases where you actually are seeing some destruction of other cells in the brain, and it ends up looking in some ways like MS. And so it's like, we cured them of one brain disease and we gave them another one. What's going on? And what they realized was that your brain is normally colonized by a whole bunch of viruses. And T cells usually are going to your brain every day to kill those viruses and to keep those viruses in control. And if you keep T cells from going into the brain, those viruses now can grow out of control and cause damage of their own. And so this is particularly related to a virus called JC virus. It's in the brains of, I can't remember the numbers of individuals, but in some patients, basically, that when you block T cells from going to the brain, suddenly these patients had PML, which was the reactivation of this virus that was in the brain. This drug has sort of gone back and forth on and off the market when people are being tested to see if they have the virus. The idea is that you could still treat people who don't have the virus, but it's still kind of not always been working super well. And so our current understanding of immune privilege is that the environment in these locations is different in terms of cytokines, so that it maybe pushes cells more towards regulatory responses, or we push towards sort of inhibiting inflammation, but we don't actually physically block the cells from going in. But what you can see is that this is all, within the past 20 years that we really understood that no, it's not a physical blockade like we usually thought. It's probably more like a turning down the response. But that does, in fact, have consequences, as we see in the Ebola case. Have an awesome break.